All right, everyone, we're going to go ahead and get started. Thanks so much for being here. For folks in the room, thanks for swimming over. It's a little bit wet out there. I appreciate it. I think our online presence is probably a bit higher right now because of the rain, but thank you all for being here. My name is Cinnamon Moffat. I am your host for this event this afternoon. This is a hybrid event, so we have folks online and folks in the room. For folks online, if you haven't been with us before, please use the chat function in Zoom to interact with us, ask any questions. Our volunteer Roseanne will read those out to our presenter today, and you'll be able to get those questions answered. For folks in the room, as you all know, just raise your hand. We'll bring you a mic, and you can ask your questions that way so folks online can hear. I wanted to make a couple of announcements, and when I started making my announcement list, there is a lot going on in the next 2 weeks. So I will take us through them, and then if you have questions, you can ask me. But Marine Science Day is our big one. We are kicking that event off on April 12th, which is this Saturday from 10 to 4. We also have an early career group that's meeting about building and updating your personal web page and website on April 14th at 10. We have our Laverne Weber visiting scientist, Marley Parker, is going to be doing a series of workshops. The first one is April 15th around field photography. If you are interested in that, please register. A link came out earlier this week to do so. We are then hosting a very special event, an evening of science and policy with Jane Lachinko on April 16th. Free appetizers start at 5 if you need an enticement to come and listen to Jane. And then the talk starts at 6. Our next seminar is April 17th, when Matt Harwell is going to be talking about building your science communication strategies. And then Marley's second workshop around elevating your value in your voice will be on April 22nd. This will be followed by a field day to kick off field safety season on April 23rd. And then Marley will be wrapping up this crazy 2 weeks of science communication blitz with a Science on Tap on April 23rd. So there's lots of ways for you to come and interact. A lot of this has come out of events that are happening around us right now and the need to figure out how to elevate and celebrate our science. And so this is how our community is kind of responding to that. I'm hoping that you have time to engage in 1 or 2 of those events, but we're excited to have that kind of coming our way. If you need information on any of those events, you can go to the HMSC homepage, go to the events page, and all the information on how to log into those. If they're on site or if they're remote, you'll be able to log in there. Or you can reach out to me and we can get more information. But Joe's actually kicking this whole thing off today. And so let me do a quick introduction of Joe Dietrich. Many of you know, but Joe comes from a background of engineering, receiving his undergraduate degree in civil engineering from Case Western Reserve University in Cleveland, his master's and doctorate degrees also in civil and environmental engineering from Washington State University in Pullman. He joined the HMSC community in 2004, transferring his research from human health and risk assessment and human pathogens to fish pathogens and environmental risk assessment involving threatened and endangered salmon. Initially affiliated with OSU Simmers, UC Davis, and commercial contractors, he has been a full-time NOAA employee with the Northwest Fisheries Science Center since 2007. While initially focusing on areas of ecotoxicology, Joe's current work with the Fish Immunology and Disease Group has been to support the research and development of sablefish aquaculture in the Pacific Northwest. And my guess is that's what we're going to learn more about today. So I'm going to hand this off to Joe. Go ahead, Joe.
All right, thank you, Cinnamon. And thank you all for coming out today. And for those that are online, I appreciate everyone's interest. And we're just going to jump in. Maybe we'll go this way. There we go. I like to start off presentations really highlighting how nice it is to work here at the Newport Research Station and the Hatfield Marine Science Center. And I thought I'd just leave this in today so that for everyone here, we can also see that all the work that I'll be talking about today was done right over here in the NAL building, the Newport Aquaculture Lab, and specifically the disease wing. if you've been to different aquaculture talks, you've probably seen a slide like this. This is from the FAO, the Food and Agriculture Organization of the UN. And it really highlights the importance of why the US and especially the Department of Commerce and NOAA are interested in aquaculture. This is a timeline series on the horizontal axis with the vertical axis indicating the amount of seafood that is produced per year in metric tons. And the blue color indicates the wild-caught fisheries, while the red and the pink indicate aquaculture. And what this clearly shows is that for the past few decades, the wild-caught fisheries have been flat, either due to fisheries management or declining stocks. But what we see in aquaculture is a continued increase at an average annual rate of about 3.2%. So how does this look for the US and North America? FAO has also generated this data for North America, and we see the same similar trend with the wild-caught fisheries in terms of that being flat over the past few decades. But we really don't see that increase in aquaculture, especially if you compare it to China, where not only do we have a change in the vertical axes with the amount that's produced, but we see that huge increase with aquaculture. And what that then works out to for the US is that we are importing about 70 to 85% of the seafood that we consume, with about half of that coming from aquaculture. And so that's big money. That's about $17 billion seafood deficit. And so again, that is the opportunity for more money coming into the US, more jobs, but we're also looking at a growing population. So additional sources of protein could be manufactured and produced here in the US. Now, this would be a good time to talk about the outline for the presentation. And when I was thinking about the different projects that we've done related to aquaculture and the different variables we've looked at, I thought I could produce a nice, pretty word cloud. But if you know me, that's not what was going to happen. I decided instead to put it into a principal components analysis and see what shook out. And I actually found this interesting to look at and talk about what I will be talking about today, as well as what I won't be talking about today. So just let me highlight a few things. Along these different axes, we have the different species that we work with. Obviously, Sablefish is in my title, so we'll be talking about Sablefish today, but we've done a bit of work with some salmonids and other marine species like herring, so they'll come into play. We have done work with some oysters, but that'll be saved for another talk at another time. When it comes to disease, we work in areas of prevention with respect to vaccines and supplements. We also look into disease susceptibility, and we'll spend some time talking about disease transmission today. So we'll kind of cover this throughout the afternoon, or maybe just the hour. So why sablefish? If you're not familiar with sablefish, they are a native groundfish species here in the Pacific Northwest, and they've had different names. In the past, they've been called black cod or butterfish, and historically, they've had a high market value with their wild-caught catch kind of mirroring what was seen with the FAO in terms of leveling out. However, in the past few years, there have been high recruitment, so there has been some change there with a number of sailfish being caught and some decrease in price. But it is still an attractive aquaculture species. And while we are a research station for the Pacific, or for the Northwest Fisheries Science Center located here, there's another Northwest Fisheries Science Center research station located in the South Puget Sound called Manchester. And they've been spending years looking at improving aquaculture and trying to get sablefish aquaculture off the ground. And they've been using net pens located in South Puget Sound and have been developing early rearing technologies, such as a non-GMO technique to have a monosex culture that grows rapidly. So fast that they can get their market size up to about 2.5 kilograms with only about 1.5 to 2 years of production. And although a lot of their work has been done on the early developmental stages, they do have partnerships now with the Jamestown Scullum tribe in order to look at actually production level decision-making and getting some of the products out to the local communities and restaurants. So why are we getting involved? Well, a big step in getting those fish quickly up to that production size is to get them from their land-based tanks where the spawning occurs and the larval development out to those net pens so they can grow unrestricted. But what they were observing was that as summer progressed and water temperatures increased, the sablefish started to present disease in these net pens. And so we were sought out and asked a pretty simple question, that they were vaccinating these fish against Vibrio, which is a bacterial marine pathogen common to salmon. They were wondering, are these sablefish even susceptible? Are we using the correct preventive strategy? And they were reaching out to us because my supervisor at the time, Mary Arcush, and I have been conducting a number of pathogen challenges where we use this assay to look at the whole organism health and whether their immune function is properly responding to a pathogen after some sort of treatment or stressor from the environment or within our own laboratory. And we're going to talk about pathogen challenge and results a lot today. So I thought I'd spend this slide at least give you some background of what you're going to expect. So they all start with, in general, some sort of treatment for your fish. We then split that treatment group into replicate tanks that will be exposed to the pathogen, as well as control tanks that are not exposed to the pathogen, but at the same stress. That exposure could occur through an injection, but we actually prefer an immersion scenario, which is more environmentally relevant. So they're exposed statically for a short period of time. We then return them to the tanks and monitor their survival over time. And then we can use survival statistics to compare different treatment levels. So when we were asked about whether these sablefish are actually even susceptible to Vibrio, we looked at the vaccine that they were administering, and it was a multivalent vaccine that contained 3 different Vibrio species. So we said, well, let's look at whether those species are actually virulent to sablefish. And these were Vibrio angularum, splendidus, and ordali. So we set up these exposures at different concentrations and then monitored their survival. And so in this upper panel, I have just an example of the results in the survival curves for sablefish exposed to Vibrio angularum. And you see at low concentrations, there's high survival. As you increase the concentrations, we reached about 75% survival or about 25% mortality. So that level of mortality is not insignificant, but for the disease that was showing up in the net pens, it's unlikely that they were able to get that level of pathogen out in the net pens. And as far as the other 2 Vibrio species, they weren't susceptible at all. So very low to no mortality. So definitely seemed like something else was present. While we were conducting these studies, they were having additional outbreaks, and a colleague was able to isolate a bacteria from one of the sick sablefish and classified it as an atypical Aeromonas salmonicida. So we got a hold of that isolate, prepared challenges just like we did with the Vibrio for that atypical strain, as well as a typical A. salmonicida that is from an infected salmon. And what we saw was something very different from the Vibrio. Again, at different exposure concentrations, we do see increased susceptibilities, but now we were seeing much greater mortality at lower concentrations. And we were also seeing that the atypical strain was more virulent than the typical. And we can see that a little bit more easily when you plot the concentration of the bacteria versus the mortality that we observed. So that top line is that atypical strain isolated from sablefish, and it's always higher than the typical at comparable concentrations. We can also note that the disease that presented is very similar to furunculosis that occurs in salmon with very localized lesions on the dermal area. We also had that isolate, had its genome sequenced just to verify that it was Aeromonas salmonicida. And here's just a quick snapshot of its phylogenetic tree, just to highlight that it is clustering with additional Aeromonas species. But this particular isolate, which we are calling T30, was most closely associated with the Aeromonas salmonicida. I've mentioned a couple times now this typical versus atypical, and that the typical is often found in salmon, but really that designation of typical versus atypical just has to do with its growth and what happens when it grows on auger. The typical strain just turns brown, whereas the atypical has no color change. So that's just that quick difference in designation there. Since T30 was initially identified and sequenced, there have been continued outbreaks, and different isolates have been collected just to make sure that there hasn't been a significant change in the pathogens that are present in the net pens. And so we do have new isolates, which I've designated here, SPEN 30, KJ1, and KTW, and they are all atypical strains and with just small variations. I'll mostly be talking about T30 today, but these three will come back towards the end of the talk. So we know that the fish are sick, and we know that it is can affect their own production at the net pens, but we are also concerned with what's happening to fish that are around these net pens. So this is the net pen located in Manchester, and the key here is that these net pens are not isolated from the environment. Yes, the fish can't leave, but pathogens can definitely be shed from inside those net pens through the net pen netting out into the environment. And therefore, if we have fish that are swimming, wild fish that are swimming around those net pens, they could be at risk from disease transmission. In addition, we can't control all the pathogens that are coming in, and we're not sure where this Aeromonas salmonicida is coming from. So we could have sick fish in the environment that are also shedding back into the net pens. And so this is something we want to evaluate in terms of what sort of risk is involved out in the environment. And to do that, we wanted to first identify what species are of concern in the area. And so there are 3 salmonids, Puget Sound steelhead, Puget Sound Chinook salmon, Hood River Canal summer run chum salmon that are all listed as threatened by the Endangered Species Act that could be in the area during their migration. So we wanted to evaluate whether they're at risk at all from this disease. We also included Pacific herring as a marine species that could be in the area that would also be of interest. So we set up a similar type design where we wanted to assess what is their susceptibility to this pathogen, Aeromonas salmonicida, but we're also interested in the carrier status. So if they are either asymptomatic or survive the infection, can they shed that bacteria back into the environment and affect sablefish themselves? So we've set up some analysis of the different tissues from the survivors and looking at qPCR to evaluate what levels of the pathogen are present. And then if they are susceptible or if they are carriers, the next step is to see is there actually transmission between species? So does a sick fish that is present among susceptible fish transfer that between sablefish or to sablefish? So this is very much ongoing in our lab right now, and I just have some preliminary results to share with you at this point. We have done exposures with steelhead at different times, and it's safe to say that they are not susceptible to this pathogen. No mortality, and they're quite robust even at high exposure concentrations. Are they carriers? This is still a potential question, or still some potential here. We have identified very few instances where tissues do have our pathogen, but at very low levels based on the qPCR, such that they are at the very bottom of what we may consider significant. So although they may have some of the pathogen present, whether they actually are able to shed that into the environment is considered unlikely at this point. With respect to Chinook salmon, again, at very high levels of exposure, they are showing no signs of susceptibility to this pathogen, but we still need to determine whether they have any carrier status. And so we've got the lab full of Chinook salmon ready to do that now. This goes for chum salmon as well. We actually just received some last week. And so we're looking forward to analyzing not only are they susceptible, but also the carrier status. We have a very different picture when it comes to Pacific herring. They are definitely susceptible and they appear to be carriers as well. And so this is a herring that is currently or is infected in this photo, and it is showing typical signs of an Salmonicida infection with characteristics similar to what we saw with sablefish. So I'll highlight that here, and hopefully you can see there's definitely some reddening in the dermal area, some hemorrhaging that's occurring, as well as a localized lesion that's forming. But then a new symptom that we don't see in sablefish is located towards the tail, and hopefully you can sort of make that out in the photo. But here we have the scales from the herring are actually exfoliating or sloughing off in response to this infection. And we do know that herring tend to lose their scales more than sablefish for sure. So we don't know if this is the fish itself as a defensive mechanism or whether it's actually a specific symptom of the Salmonicida, but it is definitely something particular to herring that we've been observing. In terms of their survival, we've run a number of just quick preliminary challenges, and we're seeing high levels of mortality, both by injection as well as in bath tests. We've been evaluating what the effect temperature has, and we're also seeing something that's a little bit different than sablefish. For sablefish, I mentioned, they start to see disease later in the summer as water temperatures warm. And really there's a cutoff. Once you get to about 15 degrees, the sablefish start to really succumb to these infections. With the herring, we're actually seeing a bit of a U-shaped distribution when it comes to temperature. At warm water temperatures around 18 degrees, we see high mortality. As waters cool to about 15, that's where we saw our initially our lowest mortality. And then as they cool further to 10 degrees, that mortality starts to go back up again. But what is, I think, really interesting at this point is even though there's low mortality at this 15-degree mark, this is where we saw the highest level of carriers. So over 50% of those fish that survived were carrying the pathogen in their tissues and at very high levels in our qPCR assay. So definitely suggesting that there could be some potential for shedding of that bacteria into the environment. And our next step would be to run those cohabitation challenges with sablefish and Pacific herring to see if there's any exchange between the two. So the fish are sick. We do, or we have in progress, an assessment of what sort of risks that is to the environment and what risks they have from the environment in contracting the disease. But we really want to control the disease in the aquaculture setting. And so I have 4 bullets up here of different control strategies, and they're not specific to fish by any means. They could be something that I'm sure you would consider yourself if you were ill or someone was ill in your area. The first on the list are antibiotics, but I'm going to cross that off our list, mainly because it's reactionary. You've already got your sick fish. We want to not deal with sick fish. We don't want that to happen at all. But also, these are open net pens, and currently there are no permitted antibiotics that could be used in the net pens. We'd rather focus on the next 3 bullets that are related to prevention. So stop the disease before it happens. The next bullet regarding biosecurity, these are best management practices at facilities. We're talking about disinfecting nets and boots and wearing gloves. And again, we're not going to focus on that today and just leave that to the facilities to just follow those practices. Instead, I'll be focusing on vaccines. And although I have a syringes picture there, we'll look at different delivery options as well as different compositions of vaccines. And in bolstering the immune response, again, we're looking at something that is not specific to fish, and that's actually adding dietary supplements, which could bolster their immune system. And it's something that you might consider, such as even the ginger that is in my tea right now. So let's start off with prevention through vaccination. Like I said, we have two variables here to consider, delivery and composition. As far as delivering vaccines, we could be looking at injection, immersion, or a bath situation, kind of like our challenges, or an oral delivery. And these bullets are arranged such that as we move down those bullets, we're trying to get to techniques that have lower fish stress and reduce the labor that's involved in delivery. When it comes to composition, we can have different vaccines that work throughout aquaculture. Vaccines have worked for viral and bacterial targets, and they can be composed of a multivalent vaccine, which means that they have multiple components of either that virus or the bacteria. We'll talk about bacteria today. They can include adjuvants, which are carriers for the vaccine, but those adjuvants themselves could bolster that immune response. The type of bacteria can be inactivated or killed within the vaccine, but you can also use live bacteria as long as those virulence factors are attenuated. So we have looked at a number of these different factors and crisscrossed them at different times. And so I'll kind of get into that, but I'm actually going to give you the punchline up front. We have vaccines that will work for sablefish in this disease. They can all be injected with a very simple formulation, but we are still striving in this research area and in this space because we want to get to those lower bullets of reducing fish stress and the labor involved in delivery. But let's start off where we started off, which was the initial disease control plan at Manchester was to vaccinate them with Vibrio species. They had a commercial vaccine. We said, well, let's start there. You have a multivalent vaccine already with an adjuvant and Vibrio species. Let's add our two Aeromonas species, the atypical and the typical, to that commercial mixture. And let's test whether we could do a bath, and let's compare it to what you were doing with injections already. With the bath, we wanted to push into a smaller size class. With the injection, we stayed at 50 grams, which is what they were doing at the time. And when conducting the vaccine studies, we have our treatments that are vaccinated, as well as sham vaccines. So, our fake vaccines, which could either be just a bath with seawater or an injection with just a saline solution, but I'll have those shams present on the survival curves as well. So when it comes to the immersion, I have that in the upper panel here with the sham treatments in black and the vaccines in gray. And what you can see here is that the survival curves for both the vaccine and the sham are within each other's 95% confidence intervals. So we really were not seeing any benefit from the immersion. The big contrast here is with the injection. The sham had about 50% mortality, whereas you injected the vaccine, this multivalent vaccine, and you had high survival. One way to delineate this and talk about efficacy of vaccines is to compare the sham to its vaccinated with something called an RPS, or relative percent survival. And so in this case, we had about an 80% RPS, which is a very high value and very effective strategy. So we have one that works, as I mentioned. But we want to move into that lower fish stress, not handle every single fish, not inject every single fish. So moving down our bullet list to an oral vaccine, we were able to partner with Oregon State University and the University of Idaho to get some funding from RAC, which is the Western Regional Aquaculture Center, to look into using alginate complex particles that Matt Hawkyard was developing here at OSU and is now moved to University of Maine. And these alginate particles could actually encapsulate our inactivated bacteria, as well as have some liposomes that would help with stimulating the feeding for the sablefish. And we could then just in a normal course of feeding, administer this vaccine. And so when the fish were about 5 grams, we started a 6-day feeding of the vaccine. We also performed an injection and then provided an oral booster about 6 weeks later, and then performed our challenge to see how well it was doing. And what we saw was that with 2 different types of particle formulations, we did see an improvement over the sham. So we did see that we could get an RPS value close to about 20%, which is definitely an improvement, definitely shows that there's some protection there, but it is nothing compared to what we were seeing with that injection, which now has an RPS of about 95%. So we have some promising results, but that injection is still the winner. And in fact, even though we are still seeing high levels, this one experiment did push that injection model a little bit further, because we were able to not only provide a vaccine that worked, which was now just a monovalent, and there was no adjuvant. And if you look at this, the fish size, we were now down to 5 grams. So it was still an advancement as far as getting those fish out to the net pens earlier with this strategy. But we still wanted this oral to work. So another technique would be to combine the oral and the bath treatments within a hybrid vaccine to see if we could then build on each one of them to get a higher immune response. So we looked at combinations of a primary oral vaccine followed by a bath vaccine, and then switch it and have a primary bath vaccine followed by an oral vaccine, and compared both of those to bath alone or oral alone and their respective shams. The lines get a little bit complicated here, but if we just focus in on the RPS values, what we saw again was about a 20% RPS in the best case scenario. So even though we have this combination, we're having comparable levels of protection from the bath or the oral or both together. So we still have significant room for improvement in order to match that injection vaccine. So where are we left and what areas could we improve? So we have been focusing on the inactivated bacteria. What about the live or attenuated bacteria? That offers another opportunity to maybe have some additional factors that can result in greater antibody production. So let's turn our attention that way. But we have a hurdle to overcome first, which is to actually generate species that are attenuated. And we were able to collaborate with the University of Idaho, who could do all of this culturing of these parent virulent strains and successfully culture them within antibiotic media in order to have those strains lose their virulence factors. So now I'm talking about looking for candidate attenuated strains from all 4 of our different atypical isolates that we have. And once the University of Idaho performed that culturing and went through the successive antibiotic treatments, we were presented with 3 different candidate species for each one of these parent strains. And we needed to test them out before doing any vaccine study. And the easiest way to do that was just to perform a mock injection, or actually a real injection, but a mock vaccination, and see if any virulence factors were carried forth or whether those candidate species were fully attenuated. And so here are just the endpoint survivals for each one of those strains. And I can just highlight what each one of these panels looks like. So for T30, we have our parent strain, and as expected, it's highly virulent, and we had no survival. Our controls are just a saline solution, so you should have 100% survival. And then we have our 3 potential candidate species, or isolates. And so what we're looking for are these 2 here as examples where they have 100% survival of this live bacteria when injected. And we saw that in 2 other candidates within our different atypical strains, and only K2W was one where we were able to achieve partial attenuation, but we still were experiencing mortality, and we don't want to have that carry over into a vaccine. So the next phase is to take these candidate species and incorporate them into the vaccines for testing both the bath and as an injection. And by now you should be probably predicting what the responses are going to be. The injection, which are these blue lines up at the top, we're showing very high survival, well into the 90+% RPS values, but the baths were not showing any improvement, or maybe just a little bit of improvement. So there's still some work needing to be done there, and it wasn't just the attenuated. But we do have hopes to maybe boost that dose up, which might be a factor, as well as some other culture conditions. So let's leave vaccines there and move to our last bullet regarding prevention strategies, and that's bolstering the immune response. And we started to get involved in this work because a colleague of ours, Ron Johnson, photoed here, has been working with alternative feeds for a number of years up in Seattle at the Northwest Fisheries Science Center. And he has been looking at replacing or reducing the amount of fish meal and fish oil that is used in aquaculture feeds with vegetable or plant-based sources, and then using macroalgae to add those essential nutrients for marine species. And he's was seeing a lot of success with Turkish towel, which is a red macroalgae, and the growth of sablefish, and was wondering if that would translate into an immune benefit as well. Meanwhile, Carla Schuberger with OSU was here and was curious about having a collaboration that would look at whether herbal supplements would also improve immunity. So as we started to consider this, we thought there are a number of different macroalgae species, and it'd be really great if we had a screening tool that could look at these different options more quickly. So I'll talk about that as well. So just to jump in quickly to the results, we looked at 2 herbal supplements as well as the macroalgae. And to do that, we had Ron actually formulate 5 different diets for us that started with his plant-based diet and then added each one of these supplements. One was curcuma or turmeric. Another one was echinacea. The third was Turkish towel, the macroalgae. And then the fourth was beta-glucan. And beta-glucan is a polysaccharide that we thought may have been the driving force behind anything related to the macroalgae. So we kind of included that as a positive control. So we fed the sablefish for 6 weeks, and then we conducted our pathogen challenge to see if there was any change in survival. So I have the herbal supplements up here, and what we see is that this higher survival was associated with fish that were fed the echinacea. So there was a improvement to disease resistance there, whereas curcuma was just matching the plant-based control. So we had no benefit there. When it came to Turkish towel, the macroalgae, again, we saw a benefit there relative to the control. But beta-glucan, which we thought would be a positive control, was not correlating with Turkish towel. So either there's a dose factor or issue there, or we're looking at a different mechanism that Turkish towel is providing the sablefish for its immune boost. So the pathogen challenge that I've been talking about today and showing here has been a great tool, and we consider it our gold standard. But you could see a study like this that involves 6 weeks of feeding and then a lot of fish in order to get our different treatments compared statistically takes a lot of time. And it would be better if we had something that was faster and didn't require fish in order to screen all the potential candidates that could be included as supplements and alternative feeds. And so we've been working on optimizing something called the Rizazerin assay, which is able to look at the antimicrobial properties of different compounds. And it's on the surface a pretty simple looking assay. It's just color-based. The chemistry of Rizazerin starts off blue, and then if there's respiration, if there's metabolism of your microbes in these wells, they start to turn color and turn pink. So you can, from this, determine whether a compound you add to these wells either is inhibiting growth by keeping the color blue or purple, or whether there's no effect and it shifts to pink. So two of the terms that we're looking to classify for our different supplements and in order to gauge how effective they are MIC and MBC. MIC is the minimum inhibitory concentration for the bacterial growth, whereas MBC is the minimum bactericidal concentration. And I have a little cartoon here to help explain that if these terms are new to you. But if we have a initial bacterial growth with this blue curve, that maybe there's no compound added to the well at all, it might have this exponential growth. And then if you add algae at increasing concentrations down here, you might see this growth curve shift downward. So when it first shifts, that would be your MIC, your first inhibition concentration. And if there's ever a curve that is completely flat, no growth, that would be your MBC. And you can look and determine your MIC from the plate just by looking at the different colors. But if you have a plate or a microplate spectrophotometer, you can actually look at this over time. And now you could have your control curve, and you compare your different wells with different concentrations of your additive, such as macroalgae, in there to see if you have any differences from that control. One thing that we did learn is that as far as the MBC, the minimum concentration where you kill all the bacteria, so 100% inactivation, it's really difficult. You can try to get that off the curves from the spec, but it's really a lot easier to take advantage of this microplate assay and just move one little dot over to an agar plate. And if a colony grows, you obviously know that you have viable bacteria in there. But if there's no growth, you can identify what is the lowest concentration that first occurs. So it's just a simple spot test. We had one more factor that needed to be optimized before we could move forward, and that was actually getting our supplements into those wells. So you really don't want to have any solid material going into the wells that could affect the color and the absorbance on your spectrophotometer. So what we realized is that we had to do some preparation, which involved drying the macroalgae, extracting it in methanol using a soxylate extraction apparatus and the tallest hood on campus, and then drying that product out and reconstituting it at different concentrations of DMSO. once we had that worked out, we're able to start testing different supplements. And in our first test set are these four here, Turkish towel, which we've been talking about already. We looked at dulse, ribbon kelp, and sugar kelp. And we are still running through the statistical comparisons now, but here's just a summary table of where we're at. And I have our MIC and MBC values in terms of grams per liter, but it's really hard to translate these grams per liter to what might actually be in the fish when they're eating the food. So I think the easiest way to look at this table is to, again, realize that for Turkish towel, we saw a benefit to disease resistance. So looking at these numbers, how do these different species compare to Turkish towel? And what I am seeing here is that at the MIC and MBC levels, we see parallel or lower numbers than Turkish towel. So these two kelp species, ribbon kelp and sugar kelp, look like they have potential for providing some sort of disease resistance or benefits to disease resistance for sablefish. So, been talking for a while, and I just want to hit some take-home messages before finishing. The first, which I already mentioned, vaccines work for sablefish, and we have a very simple injectable vaccine with a very simple composition that can be administered at small fish sizes and early life stage that will work. But we want to strive for those oral and bath vaccines that can reduce fish stress. In terms of supplements, there are feed additives that we know can bolster immune resistance, and we now have a technique to screen more rapidly for additional alternatives. And finally, when it comes to the risk of Aeromonas salmonicida transmission from the net pens, it appears that for the salmonids that we've tested, that risk is very low. But stay tuned for more information with respect to chum, which is new to us, as well as results from cohabitation with herring and sablefish. So to do all this work, we had a lot of hands, and I'd like to acknowledge the funding from NOAA's Office of Aquaculture and projects from Washington State Sea Grant and the Western Regional Aquaculture Center. And we've had help from the Northwest Fisheries Science Center, both in Montlake as well as Manchester, and their tribal and industrial partners there. And we've worked with a number of universities, Oregon State University, of course, University of Idaho, University of Maine, University of Washington, and we've been able to get our fish from different hatcheries, as well as the USGS Marrowstone Marine Field Station with herring, the Quinault National Fish Hatchery, Minter Creek Fish Hatchery. And of course, none of this would have been possible without the small but mighty group working at the Newport Research Station, including Mary Arkush, who's now retired, Mary Beth and Aaron, who have moved over to Alaska, and the RSF building, which I think stands for Reasonably Sized Fish Lab. And of course, Cam and Kelly, who are still working hard in the disease wing every day. All right, thank you.
Thanks, Joe. Appreciate that. I want to open it up for questions, and I want to start with those online. Roseanne, anybody online that has a question? Okay, for folks online, if you have anything for Joe, go ahead and put it into the chat box. How about folks in the room? Hi, Chris.
Hi, Joe. Good talk. Really enjoyed it. It's nice to see all that work put into a talk and coordinated and collaborated. So the question I have for you is, what's the current status of sablefish aquaculture in the country and globally?
So in the country, it is still dominated by the what's happening in the Puget Sound. So it's the net pens that are located there, and I still would call them as experimental net pens. The partnership with the Jamestown Scallum tribe has been able to off-sell products, and they do sell them to different restaurants along the West Coast. But the larger commercial operation is actually happening in Canada. So in British Columbia and off Vancouver Island. I think everybody is dealing, as far as in the US and in Canada, with questions regarding marine net pens. And I'm not sure what the status is for those in British Columbia, but at least in Puget Sound, they've been dealing with state regulations, and there have been legal battles as far as whether they'll be able to have any permitted net pen operations. In terms of Oregon and California, there are existing state laws that do not allow for net pen operations. So there's definitely been an interest in developing technologies to move sablefish purely into recirculated aquaculture and land-based systems. But as I mentioned very early on, a lot of the benefits come from reduced resources and labor when you can get these growing in the larger net pens. And just, I guess, one more thing globally is that one of the largest markets for sablefish is in Japan. So I think that is still strong.
So Joe, I was curious about the life stages that you've tested with the salmon, and were those relative life stages that would be exposed to the net pen?
Yeah, so the key here was that this pathogen, the atypical Aeromonas, was identified in saline waters. So that's a key component, is that all of our challenges have happened after smolting with the salmon. And we've really strived to make sure, or strive to have those challenges as close as we can to that smolting, with the idea being that the greatest environmentally relevant exposures would happen as those salmon are out migrating through the Puget Sound. So making that initial transition from the freshwater to the seawater before they get to open ocean. So that's been the window we've tried to target. And everything we've done has been on juveniles, though. So if you go back to the idea, or my slide that had the net pens and the potential for wild fish swimming around them, there definitely exists that potential for adults that are returning to carry disease as well. And we haven't studied and looked into that life stage, but that is something where it has been established in Scandinavia that you can get sea lice transfer from wild fish to salmon and net pens due to those wild fish returning as adults.
Thank you. Other questions in the room?
Hey, Joe, thanks for the talk. I'm curious about the herring and what tissues you looked at, and especially curious about the scales and whether you tested if those carried because you were talking about them being carriers, and what tissues you saw acting as carrier material and whether the scales were involved.
Yep. So we have not looked at the scales particularly or individually for infections. Our tissues that we've been collecting have been gills. We've looked at kidneys, and we've collected spleens, but we haven't looked at spleens yet. And then we've also looked at the gastrointestinal tract. So the idea being here is that with first exposure, you'd expect that there'd be some pathogen transfer across the gills into the gastrointestinal tract. And then as they start to get infected, you might start to see them showing up in the kidneys and in the spleen. And so for us, we've only been working with herring for about the past 6 to 8 months. So it's kind of a new species for us. And we've been working out some protocols, but we have seen high numbers in the gills, which I guess is not unexpected. And I believe the gastrointestinal tract is the other tissue that we've investigated, and we are seeing positives in there as well. And I don't think we've looked, even though we've collected the spleen, we haven't looked at those yet.
Has Manchester stopped vaccinating for Vibrio?
Yes, they have. And I didn't speak about it at all today, but at the production level, they are experimenting with some of these vaccines to see if the mass vaccinations do translate to higher levels of survival in the net pens as they get older and older. And so that's still a question that we have. We know that those injections work in the short term that we're doing in the laboratories, but like I said, the production level takes 18 to 24 months. So whether that protection lasts into the next year and into the next summer is still something that we're looking into.
All right, we got a question online. Go ahead, Roseanne.
How prevalent are sablefish bacterial infections in nature?
So that is something that is always hard to tell because they can be throughout the coastline, and actually getting that data is not easy. Some of the initial, well, when we were first starting with sablefish, we were wondering what they could be susceptible to when we went to the literature. There are just a few and limited numbers of wild infections that have been observed. One of which was with Rhinobacterium salmonarum, which is another salmon pathogen. And interestingly about that one is that the sablefish that had a coho salmon in its stomach. So whether that was actually the first documented evidence of species transmission for sablefish could actually be in that paper, I'm not sure. But when it comes to sablefish and these atypical Aeromonas infections, it is not something that is specific to Puget Sound. We already mentioned the aquaculture efforts for sablefish out of Canada, and they are also working on this issue with atypical Aeromonas salmonicida. So it is known as a problem for the species.
Roseanne, do you have another question there?
Any theories about why oral bath vaccines aren't working?
Well, our initial theory, or the thing that we're going to be primarily pushing for in the next round, is a matter of dose. So when it comes to the injection vaccines, it's a very straightforward dose that you have in your syringe that is directly administered to the fish, and you know that it's getting into the tissues. When it comes to the oral vaccine or the bath vaccine, it's really hard to determine which dose they are specifically getting into their system to trigger any sort of antibody production. Specifically for oral, obviously, we could track ingestion, but you do have some components of that that might just be vacated out during any sort of elimination from the fish. When it comes from the bath, it's really how much of that vaccine is being transferred across the gill membranes or into the dermal surface area. So I think dose is our biggest question here. And what we're going to try to do to increase that dose is to increase the bath concentration, but we're going to need to modify our experimental design and our vaccine design so that we can have supplemental oxygen so that they can actually survive those higher concentrations of bacteria during that challenge. And so then aside from dose, the other concern or variable that is different from injection is actually having the response of the sablefish to your vaccine, getting that vaccine to where it needs to be to produce its antibodies and respond to the specific infection. So whether the oral exposure or the bath exposure working in to out is actually getting to the tissues to produce the antibodies is still a question we need to address.
All right. I think we'll let you off the hook, Joe. Thank you very much. One more round of applause for Joe Dietrich, please. All right, everybody. Thank you for being here, and please engage in the next couple of weeks. We've got a lot going on. Hope to see you again soon.